All right. So looking at chapter 11 here, we're going to talk about chromosomes and cell division. Specifically in chapter 11, we're looking at the process of mitosis. Okay. So this just gives us a general idea of how fast our cells replicate. So how do we go from one cell to two cells and so on? And we see that things like bacteria, specifically E. coli, is going to replicate every 20 minutes. Yeast takes about 2 hours. Then we see that even within our cells, our body itself, we get different rates of replication. Our intestinal cells, which have to be replaced rather rapidly, 48 to 72 hours. Skin cells are going to be really quick. The process of making sperm is relatively fast. 64 days isn't all that long. Liver, 1 year, which is why if you get damage to your liver, it takes a long time for that recovery to happen. Human eggs are really interesting. When you are born as a female, you have all of the eggs that you will produce in your whole life. And so you're really not doing that meiosis throughout your lifetime there. And then nerves, for the most part, once those develop, you don't replicate them anymore. That whole process is done. We see that we've got slight differences in chromosomes between our prokaryotes and our eukaryotes. We see that our prokaryotic DNA is circular and it reattaches to itself, whereas within our eukaryotes, like us as humans, we have this linear DNA. And so we've got this slight difference there between these two types of organisms. The life cycle of the cell is the cell cycle, and it goes through these various processes, and we'll discuss those. And an important part of this cycle is DNA replication, which we just talked about a couple chapters ago. Because the whole purpose of DNA replication, making extra copies of that DNA, was to make sure that both cells, when we divided, had DNA in them and that they were exact copies. So why do our cells divide? We need to grow, right? At one point you were a baby. Now you're likely an adult. You're a much larger person than you were when you were an infant. Okay. So in order to get bigger, we need more cells. And so our cells will divide and replicate in order for us to grow. Reproduction itself. Okay. We're going to do this process, specifically the process of meiosis for reproduction. And we'll talk about that in chapter 12. And then maintenance and repair, right? We get injured, we get cuts, we lose cells through abrasion, whatever it might be, and we have to replace those. And so our cells are going to divide in order to replace those cells that have been destroyed due to this injury. Okay? The cell cycle in prokaryotes is simpler than the cell cycle that we see in eukaryotes. We've got this method of replication within prokaryotes that we call binary fission, where we go from one bacterial cell to two bacterial cells. So they will replicate their DNA, okay, through a somewhat similar process to what we talked about. It is a little bit different, though, because they have circular DNA. Then that cell gets longer, it grows a little bit, and then it pinches off so that we've got two cells that are exactly the same. And so we get this binary fission. This is our reproduction within things like bacteria. So we result in two cells that are genetically identical to one another. Eukaryotes, however, is a little bit different, where we've got this division because we've got a nucleus. And so we've got to do something with that nucleus before we can create two new cells. And then we've got this special process of cytokinesis, which is this process where we are separating from one cell into two cells. And this is going to differ depending on the type of eukaryote. Specifically, we're going to talk about the difference between animal cells and plant cells. Okay. So within this cell cycle, we've got two nuclear division processes in eukaryotic cells. Okay, so we've got two main mechanisms of how cells are going to divide. We've got mitosis, which is going to happen with all of our somatic cells. Somatic cells are just your typical body cells. Most of our cells are somatic cells and they will undergo mitosis. Then we also have meiosis. You might hear this pronounced as meiosis. Personally, I prefer to say meiosis. This only happens with our sex cells. So sperm and eggs. Okay, that's the only time we're going to see this process of meiosis happen, and its whole goal is sexual reproduction. And then we see that the whole process is finished by that process of cytokinesis. This is the actual cell division. Okay. Within this eukaryotic cell cycle, we have this phase, interphase, that comes before either mitosis or meiosis. So they're always going to be preceded by this interphase. And we've got three different parts of interphase. We've got our gap 1 or G1, you'll often see this written as, where this is where the cell is growing. Okay, it's getting bigger, making proteins, making sure all of our organelles and things function correctly. And then when that cell gets to a certain size and it's ready, it's going to go ahead and go through DNA synthesis. You'll oftentimes see this written as the S phase. During DNA synthesis, our DNA is replicating. So we're pairing those A's to T's, C's to G's, and we're making exact copies of our DNA. Then we jump into our third step here, gap 2 or G2. Here our cell is growing some more and it's getting ready for this cell division process. Okay, so again, interphase is going to come before mitosis and meiosis. Okay, so then after this interphase happens, we can jump into either mitosis or meiosis and we get our cell division and we end up with two cells that are identical to one another, if this is mitosis. But mitosis has 4 very specific phases that we're going to talk about as well. Okay, and we also have this cytokinesis. This is the actual duplication, the separating from one cell to two cells. Okay, and so here is that DNA synthesis. This is where we do that replication, where we have the separating at the replication fork. DNA polymerase comes in, copies that DNA 3' to 5' on the template, and so on. How are these eukaryotic chromosomes organized? So chromosome is essentially another name for DNA, but it's a specific structure that the DNA is in. So we've got 2 basic terms for this. We've got our chromatin. When the cell isn't replicating, the DNA is kind of spread out throughout the nucleus into this form that we call chromatin. Okay, and so here we can see all of this gray. This is all DNA. This is in the form of chromatin. It's just everywhere. Okay, it's like a pot of spaghetti. And then we've got our actual chromosomes. So we form chromosomes when we're going to do this cell division, because it's a lot easier to separate this replicated DNA when it's in this form of chromosomes. So we have proteins, and the DNA is going to wrap itself around these proteins so that it can take up less space and it becomes more organized this way. Okay. And so DNA packaging, if you want to check out this video, it's going to tell you about this chromatin and chromosomes and so on. Okay. Structure of the chromosome. Okay, so we've got our replicated DNA. It has doubled. This chromatid is each strand of the DNA. So this new double-stranded DNA is one chromatid. On the other side is another chromatid. So within these chromosomes, these are sister chromatids because they're identical to one another. Okay, so sister chromatids are identical versions of that DNA. And what we'll see is within this structure, on this left side, we've got one sister chromatid. On the right side, we have the other sister chromatid, and these are genetically identical. Okay, so we had this DNA replication happen, and it gives us this 2 sides to a chromosome where they're identical. And then we've got this centromere in the middle, where they are connected to one another. Okay, so they're connected at the centromere. Each side of this chromosome is a sister chromatid. Okay. So when we're looking at mitosis, this leads to duplicated cells. Okay, so these are cells that are identical. So we have a parent cell. We're going to end up with two daughter cells that are genetically identical each time we go through this process of mitosis. Okay. Again, there are 4 steps to mitosis, and we're going to be paying attention to what happens to the DNA in each of these steps, what's happening to the nucleus, and then what's happening with the spindle fibers. If you remember back to where we talked about the different parts of the cell, we talked about how we have the centromeres or the centrioles. Those centrioles create spindle fibers. So we'll pick up here in the next video.